Hi, my name is Valder Arruda, and I'm here in name of my colleagues to share with you our effort to identify risk factors that contribute to immunomodulation of the factor VIII in hemophilia A. Hemophilia A is a severe bleeding disorder that affects 1 in 5,000 men worldwide. Currently, the treatment of the disease is based on the factor VIII protein concentrate infusion. However, 30% of the patients develop an antibody to the factor VIII that in hemophilia we call factor VIII inhibitors. Inhibitors, they increase both the mortality and the morbidity of the disease. And to date, there are no risk factors, whether genetic or environmental, that can predict with certainty the development of inhibitor formation. Inhibitor treatment is focused on immune tolerance induction. This consists of repetitive infusion of the factor VIII concentrate over extended period of time, is effective in 6% of the cases, and despite of being a immune-mediated disease, immunosuppressive therapy for an inhibitor is not effective. We became interested in looking at the B cell differentiation, as shown here. The pre-immune B cells are generated throughout life from the hematopoietic stem cell. When activated by exposure to the antigen, in our case factor VIII, the cells expanded and further differentiated, generated antibody forming plasma cells and long term immunogenicity. What is important here is that the role played by a B cell activation factor of the tumor necrosis factor family called BAFF. BAFF play a critical role in the survival, proliferation, and differentiation of B cells. More recently, it became clear that BAFF levels have a increased risk for autoimmune mediated disease, and this also has been shown that the development of anti-BAFF specific therapy for lupus approved by the FDA is efficacious. The hypothesis that we developed was whether BAFF levels could modulate the factor VIII immune response through effect on mature B cells proliferation and survival. Here is the demographic of patients that we studied, as typical from patients with inhibitors. We see a prevalence in our population of Caucasians, but more important, the underlying mutation, the factor VIII gene inversion deletion, is the most common risk factor associated with inhibitor formation. And this was true by a cohort of pediatric as well as adult. A group of a member of our group, Dr. Bada Doshi, in her lab, they measured the BAFF levels in pediatric and adult populations. As you can see, in the presence of inhibitors, the levels of BAFF are increased compared with those without inhibitors. In a similar pattern was also observed in adult population with or without inhibitor to factor VIII, and this samples from adults was provided by our collaborator, Giancarlo Castema. Here at CHOP, in collaboration with Leslie Raffini, Dr. Doshi, look at the BAFF level as a function of response to the ITI, as I showed you before. In black are the patients that respond with ITI, there is a decrease in the levels of BAFF, and those who fail to respond to ITI, the levels of BAFF remain pretty much stable. In a secondary study done in collaboration with investigators from the Tulane University and Emory University, we call REACH study, we look at the BAFF levels in patients that has been treated with rituximab. Rituximab is a B cell depletion antibody that focuses on the CD20. Again, as you can see here, those who fail to ITI, their BAFF actually increases after rituximab. And those who were tolerized, the number too small for firm conclusion, but remains pretty much at the same level. Therefore, the balance between BAFF levels in response to factor VIII immune modulation seems to be consistent, not only in the presence of antibody, but as well as in the behavior in terms of response to ITI and depletion by rituximab. Went on to using mouse models to address whether BAFF could prevent the inhibitor formation. To do this, animals were initially injected with an antibody cocktail against the murine BAFF and further challenged with recombinant human factor VIII protein shown on the top in blue. As you can see here in B, the numbers of animals that make an antibody was substantially lower if in the presence of a single injection of the BAFF. And interesting, showing panel C, even the depletion of BAFF levels transient, come back at day 28, the immune tolerance induction was sustained over extended period of time. And this you can see from the BAFF, the levels of IgG, or by quantifying the inhibitor by Bethesda units. Showing here, again, in red is those that receive BAFF, and in black, those who were in the control arm. And now my colleague from University of Indiana will discuss the role of BAFF in pre-existing inhibitor to factor VIII and insights on the underlying B-cell compartment responsible for it.
We determined if anti-BAFF therapy could be effective in eradicating factor VIII inhibitors. Hemophilia A mice with inhibitors were treated with anti-CD20 alone in gray, anti-BAFF alone in red, or combination anti-CD20 and anti-BAFF in blue. Recapitulating the human data following rituximab therapy, hemophilia A mice treated with anti-CD20 had a threefold increase in BAFF levels, and this rise was neutralized by addition of anti-BAFF. The combination therapy resulted in a sustained decrease in inhibitor titer and anti-factor VIII IgG1 compared to monotherapy controls. We then identified the B-cell subsets depleted by anti-CD20, anti-BAFF, and combination therapy. Compared to the monotherapies, combination anti-CD20 and anti-BAFF, again shown in blue, had a robust depletion of splenic and bone marrow plasma cells, plasmablasts, and splenic marginal zone B cells. We observed high BAFF receptor expression in germinal center B cells, whereas TAS-E was highly upregulated in plasma cells and plasmablasts, both in the periphery as well as in the bone marrow. We confirmed that combination therapy with anti-CD20 and anti-BAFF acts by depleting plasmablasts and plasma cells. To do this, we compared our treatment regimen with anti-CD20 and TACI-FC, which is known to neutralize BAFF and APRIL cytokines and impact mature plasma cell survival. Treatment with either anti-CD20 and anti-BAFF or anti-CD20 and TACI-FC durably reduced established inhibitors in hemophilia A mice. This was associated with a lack of antibody-secreting cells in both spleens and bone marrow of mice given either treatment. In summary, our model proposes that CD4 T-cell help under co-stimulation and high BAFF levels leads to factor VIII specific memory and plasma cell formation. Neutralizing plasma BAFF at initial factor VIII exposure can lead to factor VIII tolerance. Further increases in BAFF caused by rituximab-mediated B-cell depletion results in a failure to induce factor VIII tolerance and increases the factor VIII specific inhibitor response. Downregulation of BAFF during ITI, in turn, can result in tolerance by suppressing plasma cells, which, combined with rituximab-mediated memory B-cell depletion, causes sustained tolerance. In conclusion, we find that BAFF is a potential harbinger for ongoing humoral immune responses to factor VIII in humans with hemophilia A. Preventing humoral immune responses to factor VIII with anti-BAFF is effective in hemophilia A mice. Dual anti-CD20 and anti-BAFF therapy is superior to individual B-cell monotherapy for anti-drug antibody reversal. This acts via a combination of memory B-cell depletion and deprivation of factors that sustain plasma cell survival. The novel role of BAFF in alloimmune diseases requiring protein replacement can finally serve as a potential target for other diseases complicated by anti-drug antibody formation, such as Gaucher or Fabry disease.